Okay,
Great.
welcome everybody to our EV Journal Club. Today we are going to discuss a very important topic. As you know, one of the most promising application of extracellular vesicle is a biomarker, but still we are struggling with several limitations. Like, for example, purification, characterization. Still, the lack of a standard protocol is a very big obstacle that is actually slowing down the clinical translation somehow. Today, we are very happy to have two experts in the field. I will introduce Danilo, which is a PhD student from Tallinn University, also scientist at ANSA Biomed, which is a biotech in Estonia dedicated to providing solution for EV characterization, isolation, purification. And then we have also Natasha Zarovny. I don't know how to introduce you because you are involved in so many projects. So for me, it's very hard to give a background about you. I just know that you are innovator in the EV field, pioneer in the EV field. So probably you can tell us a little bit more about yourself.
Well, I will kind of compress the time introducing both the work and myself. So I've been transversally operating in this field over 15 years, uh, like bridging from academia to small and not that small industries. So I'm very much keen to issues that are really fundamental for a translation. Okay, so once that we really have a great fundamental science, what we actually need to make it as robust possible, as appealing possible to translate. Therefore, I've been in a lot of translational projects. Now work also with EV startups, and one of the projects I work with that actually initiated a little bit this idea of this work is a Nexus transition project from EIC scheme, which is actually a product development project. So if I may say immediately, thanks to Marco, to Jotbody, for shedding some light of this kind of work, and especially to Danilo, that is a PhD student that was fundamental in this work. He's also working in industrial setting, and he not only embraced, but also brought further and inspired this effort that is not to be intended only as a summary of over a decade-long efforts and hurdles in translation of biomarkers on EVs, but to bring some awareness of that this phase of setting up the specifications and requirements and design constraints is a very early step in any translational process and product development project. And it was very useful for us. We implemented this in our projects, mine, but also those that are run by Danilo in Hansa Biomed. And therefore, we hope it's going to be as useful to you as it was really useful and obligatory for us. So I would say that and leave the word to Danilo.
Okay, so thank
Thank
you
you.
very much for your introduction. And please, Danilo, you can share your slide.
Mhm.
Happy to learn more from you.
Yeah. So thank you for this nice introduction, and thank you for the invitation and the opportunity to present some of our published work. I hope you can see the slides, right? Correct screen. Yeah.
Yes.
Yes. Okay. So actually, for today, we are gonna cover in this presentation the two articles basically that we had recently published in ICER journals. Why? Because they pretty much cover the similar topics and address similar issues. Although one of them is a perspective review article, the other one is a technical report. They focus on this heterogeneity of EVs in complex biofluids such as blood plasma. They address their abundance, so the proper quantification, the relative ratios between different nanoparticles and EVs, the marker abundance and distribution across the nanoparticles, and also what kind of implications this has on the downstream analytics. So what we can expect to be the limitation or the advantage of certain analytical platforms, And in this technical report, for example, we also made some methodological optimizations and adjustments that allow for a little bit higher or actually advanced quantitative fluorescence output from our analysis. So why in general we would find blood-borne EVs interesting? Well, first of all, blood is a well-established biofluid for clinical application. It has been so for ages. It contains diverse and complex population of EVs that originate from pretty much all of the perfused cells and tissues in our body. And it's exactly these blood-borne EVs that carry this huge diagnostic potential because they offer this systemic overview of dynamic and also real-time physiological and pathological processes in our body. But before we can actually think of translating this potential into actual clinical implementation, into clinical setting, we need to consider certain questions, certain aspects, and to make these considerations beforehand. So the questions would be, what is the concentration of the plasma EVs that we have in plasma, not only in total abundance, but also relative to other nanoparticles? What would be the concentration of some disease-specific subpopulations, for example, the proportion of EV biomarkers that are carried in or on top of the EVs relative to the total plasma proteins and nucleic acids that are present in the soluble form. How are these molecules distributed among individual EVs? This is kind of useful for some advanced analytics. And finally, all of these, what implications and requirements they impose in the downstream analytics. So what we could gather from the available information in the literature is that EV concentration in plasma usually gravitates around 10 to the power of 10 EVs per milliliter of plasma. This is, of course, coming from a greater dynamic range, given the different analytical methods that are used in different articles and also the different isolation procedures. But we can say that this is somewhat an average or at least a geometric mean that is being reported the most. And it doesn't surprise that actually most of these EVs come from the blood cells themselves, because blood cells actually comprise the in count the highest number of cells in our body, more than 90%. So more than 99% actually of the total plasma EV population comes from the blood cells in different proportions, of course, and only less than 1% comes from other tissues and cells in our body. And on top of that, we can also expect to have some of the EVs coming from commensal bacteria that come from the skin oral gut axis as well. Certain estimations could be that roughly they contribute to up to maybe 10% of the total EV population. So in this whole population of EVs, then we can ask how many of the tumor-derived EVs we can expect to have there. And in order to start answering these questions, well, we have to consider, first of all, the tumor size, because different cell masses will produce different amount of EVs. And in order for us to contemplate some meaningful, at least diagnostic assay in the downstream application, we would think of some tumor size that is relatively small. So something between 1 millimeter or 1 centimeter. And these tumor sizes should have roughly 500,000 or 1 billion cells comprising them. Based on the information, again, that we have in the literature, we can have an estimate of the secretion rate of cancer cells. This is roughly 2.5 EVs per cell per minute. And of course, this comes from a greater dynamic range, which is just this is just a geometric mean, which actually quite well correlates with the mitochondrial metabolism of the cell. So we could see this in the publication in the Journal of Extracellular Biology by Aubert and Sveningsen. And on a third factor that needs to be included in this calculation is the mean residence time, which is roughly 40 minutes. This is the time that EVs spend in circulation once they get released from the cell until the end where they get taken up by some other cell. When all this is calculated in, we can get roughly 10 to the power of 4 to 10 to the power of 7 EVs per milliliter of plasma coming from these tumor sizes. And once we put all this on a relative scale compared to all the other EVs, the total EVs and the blood cell-derived EVs, we can see actually that these EVs coming from the early stages of the tumor development, they can be quite diluted, so less than 0.2%. And not only that, we have quite a lot of other non-vesicular extracellular particles, which can be lipoprotein, viruses, some of the newly discovered also exomeres and supermeres. We have soluble proteins, although they are not, let's say, NVPs by MICE guidelines. I would like to include them here just because certain molecules can be actually 8 or to 15 nanometers, which actually makes them quite large if even a couple of them just aggregates together. And aside from biological non-vesicular extracellular particles, we also have other contaminating particles coming from environmental pollutants, such as micro and nanoplastics. And the problem with these all particles is basically that they interfere in our analysis on both biophysical and biochemical level, because again, they have quite similar properties in terms of size, density, and some of them even biomolecular composition is very similar to EVs. Just to give an example, lipoproteins that are probably one of the most abundant NVEPs in plasma, they outnumber EVs by 5 to 6 orders of magnitude. And fortunately enough, most of them are HDL nanoparticles, which are small enough not to be detected by single particle analysis because they are 5 to 12 nanometer in size. But unfortunately, they do carry some of the molecules that are also attributed to EVs. So that can be a kind of difficult to analyze in the bulk assays in the downstream analysis. And also we have larger lipoproteins that are large enough to interfere also in single particle analysis. And although they are the least abundant among lipoproteins, they are still more abundant than EVs, even in the fasting conditions. So they can be up to 10 to the power of 13 particles per milliliter of plasma. And not only that, we know that a lot of these lipoproteins do form this dynamic shell, this biomolecular corona on top of EVs, which actually makes a proper separation and analysis of pure EVs from plasma quite difficult. And now when we actually make the whole particle overview in the plasma, we can see that with respect to lipoproteins and other NVEPs, EVs and particularly tumor-derived EVs are very, very diluted. This is like less than 1 per myriad in the total particle pool in the plasma. And of course, we're not only interested in particles when we are analyzing our sample, we want to know the molecular content that EVs carry. And we can also make a comparison between total protein or nucleic acid content in plasma versus what EVs carry as their own cargo in this total molecular load of plasma. So EV proteins, for example, comprise roughly 15 microgram per milliliter of plasma, which is around 0.02% of the total plasma proteome. EV-RNA are actually a bit higher in this relative ratio. Roughly 18% of the total plasma RNA can be found in the EV cargo, because this is the nucleic acid that is usually protected inside of the vesicles, so it's not easily degraded by enzymes. On the other hand, we have EV-DNA that is usually carried on the surface of the EVs. Therefore, the one that is inside of the vesicles is very scarce in the amounts. So 0.005% is what we even saw in our own measurements. And if we just want to make, for example, the protein concentration overview, because proteins are usually what we analyze the most in our EV preparations, on a relative scale compared to, let's say, 3 most abundant protein sources in plasma, which comprise 99% of the total plasma proteome, we can see that EVs and some of the, let's say, maybe cancer markers that could be carried by EVs are again very diluted. This GRP78, for example, is taken because we found, for example, in one study that 1 microgram of tumor-derived EVs from cell culture can contain roughly 143 femtograms of this particular molecular species. So this is roughly 100 millionth part of the tumor EV proteins. And if we calculate into that also the total EV concentration that we could have from different tumor sizes, 1 millimeter to 1 centimeter in size, we actually see that this protein concentration can be even in femtogram or attogram ranges. So this is translating to a number of molecules. Actually, this is roughly 15 to 30,000 molecules per milliliter of plasma. So it can be almost impossible to detect in these lower ranges. But what can we do in order to at least try to ameliorate some of these issues? We can think of some upstream procedures and methodologies that would allow minimal artifact creation when we process plasma, not to create, let's say, artificial platelet vesicles by activating platelets during the pre-clearing procedures. We can think of different EV purification methods and enrichment procedures that could enrich our subpopulation of interest. And we can also choose proper downstream analytical tools that would allow maybe signal amplification if we are dealing exactly with low marker amounts, or tools that allow higher sensitivity detection or higher dynamic range. This comes handy when you're dealing with detecting particles that are in a low relative ratio compared to the total nanoparticle pool. But EV diversity and dynamicity is also affected by other factors such as inter-individual variability, cell tissue origin, whether the sample is collected in the morning or afternoon, before or after meal, in health and disease. In this particular case, I just took out a couple of figures to show the example. This is from a Newman et al. Publication from 2021. Here we can see the diurnal variability even for generic EV markers such as CD81, and also for some more specific cell tissue specific markers like this liver marker. But not only that, we see the relative changes in the abundance, like a bulk amount of these markers, but we also see them changing on the individual particle level. So the marker is differentially expressed on a single EV depending on the time of the day or fed and fasting conditions and all the other factors that we just saw in the previous slide. So this actually prompts us to think what could be the distribution of these molecules between single EVs. How can we imagine a molecular cargo of one vesicle or whole population of EVs being distributed among them? And in this diagram, I think it summarizes nicely all the hypothetical solutions that we have. On y-axis, we can see the prevalence, meaning the amount of the marker present in the whole EV population. And on the x-axis, we can see the concentration, meaning what is the amount of the marker in a single EV. So for example, if we look this left upper quadrant, we can imagine a scenario where this is like many EVs expressing fewer epitopes or fewer copies of a single molecular species. And on the lower right, it's the opposite. We have fewer EVs expressing many copies of a single molecule species. So, and anywhere on this blue dotted line, we can imagine that the total marker amount would be the same. It's just its distribution would be different. So for example, here we could have 200 EVs with 5 copies of a certain marker. And here we have the opposite, 5 EVs with 200 copies of a certain marker. So this is actually the interesting part for some advanced single particle EV analytics. But this is again hypothetical, let's say, modeling where we can see how these molecules can be distributed. But if we want to understand how many molecules can we even find in a single EV, if we are looking at different proteins, messenger RNAs or microRNAs, we can again resort to even some modeling based on the cell to EV extrapolation. So we know, for example, one single average cell, how many proteins and nucleic acid it might have. And we can just scale this down to a volume and a surface area of a single particle that is 30 to 150 nanometer and get certain number of molecules that this EV can contain. On the other hand, we can also take an average protein or an average sized messenger RNA, microRNA, calculate their size in terms of volume and surface area that they can cover in physical space. and then just see hypothetically or theoretically what is the maximum capacity of a single EV that is size 30 to 150 nanometer. And of course, this is a little bit more idealistic scenario where EV is entirely occupied in its internal volume or surface area by these molecules. And this is, of course, not realistic. It just shows us the actual maximum capacity that a vesicle can have. And of course, empirical evidence suggests something that is much closer to that cell to EV extrapolation model. We can see based on the single particle analysis of surface markers that we can indeed have a couple of hundreds of protein molecules of different kinds on the surface of PV, while on the other hand, RNA molecules can be present in one or less than one molecule per single vesicle. And if we are further looking into specific molecular species or how many copies a single molecule can have in a single EV, that dynamic range is also quite high. We can have certain molecules that are expressed in hundreds of copies per EV. This is the case, for example, with some tetraspanines, while in other cases we might have one copy per 1 million EV, and that can be also the case with certain cell-specific protein markers or RNA molecules. And not only that we can have these markers that are very rare, we also have the combination of different markers per single vesicle that can be also quite rare or even undetectable. Even if we look just at the generic EV markers like tetraspanines, those subpopulations that express tetraspanines in double or triple configurations are quite rare. And if we go even more specifically into some cell or tissue-specific markers, the double or triple configurations are basically non-detectable. So we can infer from this that subpopulations expressing many copies or at least one of these markers are actually much more abundant than those EVs expressing multiple molecular species at the same time. And this can actually bring us to some contemplation about analytical requirements that we might have here. So coming back to that diagram of the prevalence and concentration, imagine having a protein that is highly expressed, some generic EV marker. It will obviously have high prevalence, meaning a lot of EVs will express it, and also high concentration, meaning it will be expressed in many copies per single EV. So we will probably not have that big of a problem detecting these EVs. But on the other hand, if we imagine early stage liquid biopsy scenario, we will probably have something that is on the opposite side of this diagram. So low number of EVs derived from tumor-derived or tumor tissues, and low marker prevalence and concentration, which would actually require quite high sensitivity to detect those low molecule concentrations in single EVs. And also it will require high dynamic range to detect few vesicles in total pool of all the other nanoparticles. And just to give you a context, how big of a dynamic range we're talking here, well, if we just take the data that we showed before, that for the tumor-sized 1 millimeter to 1 centimeter, we can have 10 to the power of 4, 10 to the power of 7 tumor EVs per milliliter of plasma. And if we, for example, take one high-throughput instrument, such as nanoflow cytometer, that can process 10,000 events per minute, we still realize that for detecting 300 tumor-derived EVs, it would take anywhere between 15 minutes and 20 days. And of course, if you want to get a more robust statistics, get more tumor-derived EVs detected, this time will probably scale up quite significantly. So in this case, we can even consider that dynamic range can be even a limiting factor, and we might have to choose some other analytical approach or to enrich for a specific subpopulation before going forward with the analysis. And not only that, we also can talk about limited sensitivity of many single particle analyzers. This is coming from our own results from the tech report that we had in the ISEV journal. So here we analyzed plasma EVs from two different subpopulations. So from platelets and from red blood cells using CD41 and glycophorin A antibodies respectively. And we analyzed them with nanoflow cytometer and fluorescent NTA. what we saw is that, of course, non-FCM detected more because it has a more sensitive APD sensor, but it also detected more of red blood cell-derived EVs in comparison to the platelet EV subpopulation, while on the other hand, FNTA not only detected less total amount of particles, but also the proportion of this subpopulation was a bit different. There were slightly more CD41 or platelet-derived EVs. Consider that this is a logarithmic scale on the y-axis, so even the tiny differences are actually quite significant. And once we actually took this data and compared it to other published results, also fortunately that year also we had this article from JEB that we could compare to. The authors used apogee calibrated flow cytometer to analyze pretty much same plasma EVs using same markers, and they even saw less total number of particles than FNTA. And in this case, the relative ratio between platelet and red blood cell derived EVs was even more pronounced. And it makes sense because later on we saw that this instrument was calibrated for a much larger size ranges. So it probably excluded quite a lot of small and dimmer events. And this also indicated that changing the sensitivity does not only lead to decrease in the absolute abundance of the particles that we see, but also it changes the relative ratios of different subpopulations that we can detect. Why? Well, because of exactly these stoichiometric issues. So the EVs from red blood cells apparently had very few glycophorin A expressed on their surface. Therefore, they were much more difficult to detect. They fit exactly in this scenario. High prevalence, meaning they are present in high number of particles, but the concentration of the marker per single EV is actually very low. On the other side, we had CD41 platelet EV subpopulation that had a decent number of molecules presented on the surface. Therefore, they were detected more readily. So this is something that we also confirmed with the bulk analysis, that indeed glycophorin A is the predominant marker, and the red blood cell EV population is more represented in the total plasma EV pool. And again, this is not the only issue that we encountered with it. We also saw that, for example, I like this figure very much because it shows some of commonly used cytoplex and cell stream flow cytometers that were also calibrated for a very specific size range and also fluorophore load range. Therefore, they actually excluded quite a lot of events, even 90% of the events that would otherwise be detected with the instruments such as single molecule flow cytometers. So indeed, the sensitivity is a very much limiting factor, and also the calibration ranges that instruments have can also include quite a lot of limitations in that case. And when we consider that sensitivity may be an issue, we can always resort to bulk analysis, because bulk assays are actually more established. They have higher sensitivity, high dynamic range. The SOPs are well developed. We have a lot of reference material that allows us absolute quantification for many different analytes. And here we can see in this table some of the commonly used bulk analytical tools and their respective and also respectable limits of quantification in terms of the molecules that they need for a proper assay. We can see that some of these tools are quite capable of detecting those low marker, low EV scenarios that we had for, let's say, early stage tumor development. We also can see that many of these methods for bulk assessment for single molecule analysis in digital mode were also reutilized for single EV analysis. So this also shows that they have quite high versatility, so you can always play around with the methods that you have and try to get most out of it. But finally, all of these bulk methods, they finally provide only the bulk signal. So we do not really get the answer to how many EVs we have there, what is the analyte per EV. So the distribution of the marker that we might be interested in is completely not available by these methods. And also even whether the analyte is EV specific or not. So finally, we need single particle analyzers for this. They not only provide this high resolution and specificity, but they also allow us to run multiparametric analysis, which can actually lead to this multidimensional correlation matrices where we can get actually much higher analytical and informative value out of single sample analysis. For example, this is where I see that the single particle analysis having the advantage over bulk assays. And exactly in this just simple example, we can see that, for example, in a 3-parametric analysis by checking the particle concentration, the size, and the marker expression per single EV, we can easily start distinguishing the population or the subpopulation that is more distinct to the cancer cell-derived EVs. And by gating our analysis only to those portions, we can increase the specificity of the assay quite a lot. In this case, cancer-derived EVs in comparison to healthy EVs had actually larger size and fewer molecules. So this is quite a valuable information to have if we want to have a specific detection of cancer EVs. But unfortunately, most of these instruments with single molecule, single EV resolution, and high sensitivity are not yet widely available. And most of these are like custom-made tools. And finally, also fluorescence calibrators for the EV analytical range specifically are not yet available. They're lacking. So finally, what we can do, we can actually think of a solution that is maybe a combination of the best of the both worlds. So use both bulk analysis and single particle analysis to basically get each other to complement each other's limitations. And just to give you a last example here, from our tech note, from the tech report that we had in JX Bio. So we also analyzed colo EVs labeled with anti-CD63 Alexa 488 antibodies, and again analyzed with nanoflow cytometer and FNTA. And we saw again that nano-FCM, of course, detects more particles because there were less antibodies presented on the surface of the particle, so the events were dim, and not all of them could be detected with FNTA's CMOS sensor. But then again, we weren't sure whether this is even that even nano-FCM detects all of the analytes that we have there. So we analyzed it also with a bulk in a bulk signal with a plate reader, basically. And we noticed that indeed there were quite a lot of analytes that plate reader could detect in this bulk mode detection that were obviously not detectable by neither nano-FCM nor FNTA. So in this case, actually, we see how bulk measurements can help us to understand the limit of detection of our single particle analyzers. And we can definitely use this to complement bulk and single particle platforms to basically get the more comprehensive results in the end. So with this, I will just conclude with some final remarks and some considerations for everybody that maybe wants to do this kind of analysis, that we always need to keep in mind that the complexity, to understand the complexity of the sample that we are analyzing, all of the particles that are present there, and all of the molecular components that we might have interfering. To be aware of the EV diversity and abundance, and also the biomarker prevalence, concentration, and stoichiometry based on these diagrams that we showed. To acknowledge also the analytical capabilities and to use this to make informed decision for assay design and validation. Once again, we can use both bulk and single particle analysis to complement each other, which can provide an optimal solution for the best analytics possible at the moment. And finally, at least this is also my opinion, that the holistic multiparametric approach in EV analysis, which actually leverages both biochemical and biophysical information that EV convey, could indeed offer the greatest value, which we can finally translate into more potent diagnostic tools. So with this, I would actually like to thank you again. Thanks to my supervisor, Natasha, and to all the authors that contributed to making these two articles. And of course, for all of those that are coming to ICEV in a couple of weeks, I see you in Vienna then.
Thank you, Danilo. This was a very great presentation with a lot of number that definitely leave room for discussion. So if please, from the audience, any question for Danilo, you can write in the chat box or just ask directly.
So can I ask a question first?
Sure, thanks.
Okay. So how did you calibrate? How many antibodies do you have bound on ELISA test?
In ELISA, we didn't do that. So for this, if you're referring to this one,
Yes,
right?
yeah,
So this is actually, we just take the same number of particles that we are analyzing in nano-FCM or FNTA, and we analyze in suspension in a plate reader, and we use soluble antibodies in serial dilution as a reference to estimate how many antibodies we have in our own sample. So this is kind of direct actually assessment of the fluorescence based on the antibody fluorescence intensity, not the the ones that are bound on the surface of the ELISA plate.
Okay.
Yeah.
Thank you.
More question in the audience? I have a kind of provocative question for you. So basically, how reliable are the data that we are reading today in so many publications coming out every day? How reliable are it?
I guess the more we have them, the better, because we can at least make some averages out of it.
Okay.
Yeah, and indeed, I mean, reliable, what does it mean? I mean, the data can be reliable because if certain instrument is calibrated, it will probably give reliable data. But whether it's close to the truth, to the actual absolute amounts that we have there, that's the question I think that we need to ask. And this is exactly, I think it was in this particular figure, we can see all these instruments here are calibrated. So cell stream, cytoflex, they show their calibrated ranges. They express the signal in picoerythrin copies per particle, and also they detect a certain diameter range. But this calibrated range is definitely not enough to detect most of the particles that the sample contains. So finally, yes, they are reliable, but for this, like a couple of % of the particles that they detect in the end. And of course, the truth is that you have 90% of the particles that are not detected at all. So that's kind of the problem, I think, in most of the articles that we read today, what actually we are not detecting.
Yeah, that's please.
I may just add on this that indeed, I think that we have, when you say reliable, it probably also means confirmed, like independent confirmations. These things are still rare in the field, and that is what also shows a little bit of, like field immaturity. You can't expect this to be done only by scientists. That is done also by those that are translating that into some kind of like services or product pipelines. And that probably you have these initiatives like MatVes or, like efforts. Danilo also, it's here, for example, on Apogee data on bigger particles that were a little bit more feasible to calibrate with what was available. I do respect very much these metrics and standardization initiatives in the field. Although, as Danilo was saying, it's also fair to recognize that these methods and these calibrations, these instruments, are actually meant for slightly bigger particles, and that we still have this black matter that is totally missed, and that maybe we will be able or soon or not be able ever to detect reliably, but it's always useful to make a ratio of what you can detect or not, and this is already a very good additional diagnostic input. So even if you can't quantify it with something like single molecule based, At least you know that what I can detect is this, and what I am still missing but is there, okay? And let's wait for novel technologies to like make light on this.
Indeed, and actually, that could be a problem if we try to develop some EV diagnostic assay that indeed relies on this very distinct calibration ranges. For example, in this is the article that uses quantitative single molecule localization microscopy, and let's imagine that this microscope was calibrated for marker presentations of only 50 and higher. So that one would actually cut off all of these EVs that we consider cancer specific, because in this particular case, it shows that those larger sized EVs coming from the cancer would actually express fewer molecules. So we actually needed, in this case, higher sensitivity to detect those EVs specific to the cancer population. Yeah.
Especially if we consider that I've been reading many recent papers, or even biotech are focusing now in early detection of biomarker using EV. So in that case, the sensitivity should be even higher. That's a little
we never translated this to this range of early or like in asymptomatic, like patients detect something that is really very early that we can model, because we can model it not in humans. It has been modeled in animals, very few studies. We also used for this work, valuable ones, that tells you that it could be even more feasible than for other circulating biomarkers, which is what is a big drive for EVs. So you could still have more EVs to fish out than other markers like cell-free DNA at this early stages. But okay, it's really fair to you use also proper how to say wording and vocabulary. And I would also add, I think this is also provocative. We've been talking now about the digital techniques that go to a single vesicle level, and also using a single molecule technologies to detect vesicles. Maybe, and we talked about this with Danilo a lot, it would be also fair to, if we want to use properly with the highest LOD or LOQ that these single molecule methods have on EVs, then we may have to sacrifice the integrity of EVs. That's the point. Okay, reduce them to single molecules, and then we recalibrate the system and speak a little bit different language. But that's kind of one of our final thoughts after this work.
Okay, that was great presentation. More questions from the audience?
Well, if no one has a question, I have more.
Please go
Sorry,
ahead.
Danila.
No.
Just the food for thought. So first of all, tumors, solid tumors, they produce large oncozones. So maybe limit of detection doesn't need to be that low in respect to size. So some of your experiences with that would be interesting to hear. And another thought is that have you considered just filtering out, not actually filtering, but affinity isolation of vesicles from very large volume of plasma, like affinity column or something like that, not just what can fit in ELISA or these single methods. So if you make a column with antibody specific to something, and then you pass, I know, hundreds of milliliters of plasma, would it be more efficient. Yeah, I know it's a very old and rough method, but you know, that method can be upgraded. For example, instead of antibodies, to use aptamers in large quantities on some surface that are long, that can, you know, like some sea creature, just to pick up from the big volume different kinds of I mean, single kind of tumor EV or something.
Yeah, it definitely makes sense. I mean, this is exactly one of the approaches that a researcher can consider, is exactly this, to either scale up the volume of isolation and do a more specific enrichment of subpopulations that is needed. And in this case, it could be done with this affinity approach. And then again, it also depends on which kind of markers or analytical output we want to achieve in the end. For example, capturing with antibodies, I guess eventually it would mean eluting in a very harsh condition, and then we would get the eluate that is useful now for just bulk analysis, like targeting specific analytes as a single molecule and just detect it like that. But we won't, I think at least, won't be able to analyze in this like single particle multi-parametric approach anymore once they get eluted from the antibodies. That's one thing. And for the first one, for the large oncosomes, indeed, they can be larger and therefore easier to detect as a single particle in any sort of instrument that has a decent sensitivity for those kind of measurements. But here we are now facing the dynamic range issue, because we will probably not have that many oncosomes released from a single cancer cell, or at least we would expect that, you know, there would be a couple of maybe large oncosomes, but you would have definitely much more smaller EVs released. So in terms of number of particles that we then have in this pool, we would have less oncosomes and more of those smaller particles. So oncosomes would require that higher dynamic range in order to detect them if they are present in, I don't know, one oncosome per 1 billion or more other particles. So yeah, it's kind of like this trade-off. What are we targeting, sensitivity or dynamic range? It's definitely a food for thought. So yeah, I like these kind of things, and it always you know, leaves a room for questions, for thinking of an actual setting that we want to have in our study. Yeah, I like that.
Okay, thank you.
And can I also add up on this? Okay, on oncosomes, I think we have also, I mean, one has to think of that not all our plasma preparations are actually allowing us to see large oncosomes if they are there in the sample. Biologically, they might be linked to certain tumors or certain phases of tumor growth, much more like a, you know, like release of cells, because this is high material expenditure, you know, like really something that big. So probably early phases of relapse or a disease would not do it. And especially I wanted to say, yeah, that's an excellent comment on EV enrichment by affinity. This is actually the core idea of Nexus project, which is, you know, like the thing that we have to do this in situ enrichment in order to enable the analytic sensitivity. And the Nexus project also had that problem of releasing intact EVs upon affinity capture, and this is something that we provide. Okay, so just to say, I mean, this is a European funded initiative, and we will also defend this data just for people to know that this is possible. So there are specific developments that are aimed at resolving these questions, you know, because they are still questions, not widely available, as Daniel was saying, for a lot of cool tools and devices, but let's make them more widely available.
Yeah. Yeah, great. I just imagine these affinity methods as a lot of very hairy brushes put in the sample. Yeah, you know, just you sample, your sample passes, all these brushes which are hairy with affinity, you know, aptamers or something, they collect whatever collect. You remove all those brushes and put in some detection instrument. Yeah.
Yeah, but you know, like think also logistics and operational practicality of something that has to be high for output. It has to be like, and it probably, if you work with such a large volumes, then you disrupt pre-analytics in routine healthcare. And you know, we think of all
I know, it's
kind of things
complicated.
before doing it, because then we will come to the user and he will say, no, too much.
That's completely other story.
Yeah.
Okay, so that was great, Journal Club, and amazing presentation, Danilo. Thank you very much, Danilo and Natasha, for your contribution. I will see some of you in Vienna, and hopefully every one of you for the next Journal Club. Thank you again, Danilo, Natasha, and everyone.
Thanks, Marco and Danilo.
Thank you, Danilo. You were great.
Yeah, he was.
Bye-bye. Bye-bye.
Bye-bye.
Bye.